back to Young Engineers of Today. I hope you had a good couple of days, and I hope you're ready to continue with synthetic biology and all that good stuff. Um, Near as I can tell, we actually only have a week after this. We have today, we have next Monday, and we have next Wednesday. So we'll fit in as much as we can. Next Wednesday will probably be reserved for something a little bit different, or quite a bit different than what we've been doing normally, but we'll see. Uh, Either way, hopefully, we'll keep this as. No, I did get in contact with Mr. Dubik, but I haven't heard anything back yet. So we'll find out hopefully soon. Yeah. Um, so we'll see. Um, and honestly, like I was saying before, it's entirely probable that just the winner of the bridge contest gets bragging rights. Just like, oh, hey, you won. Congratulations. you know, which is not, not exciting, but at the same time, it's kind of nice to get that validation. So anyway, we're going to continue with synthetic biology, biotechnology, and bioengineering. And last we met, we got as far as edge detection. We actually went over a number of things. We talked about how there is sort of an analog between circuits and genes, well, and biology, I guess. So we can use biological systems that emulate like electrical circuits to create biological systems that operate a lot like electrical circuits. Also, we talked about the idea of synthetic biology, which is the design and engineering of simple biological systems, that aren't found in nature. We'll just start out with that. But yeah, that's sort of one of the basic tenets of synthetic biology is equating biological systems to electronic circuits. For example, fluorescent proteins could be like light bulbs or LEDs, and transcription factors could be like transistors or logic gates. And it does, in fact, simplify the process of creating these more complex biological systems because we're able to put them into terms that we already sort of are well versed in, relatively speaking. We're not so well versed in creating life, however, in the synthetic biology sense, however, we are relatively well versed in creating circuits. So being able to discover purposes for genes and proteins and stuff like that have similar purposes in circuits, it allows us to spend the time to actually create these life forms. Now, there's more to it than that. Obviously, there are a whole bevy of genes and mechanisms and things like that exist inside of living organisms that do not have any clear analog to electrical circuits. That we build. Like, we don't need to build an electrical circuit that's self-sustaining and can turn organic matter into energy and stuff like that. That's not generally something that you have available to you. But yeah, so that's more or less what we talked about on Monday. The example that we had was the repressolator, which is a set of genes that turn off other genes, repressors, all linked up in such a way that they only express themselves they go through the rotation and express themselves periodically, but one of them is linked to a green fluorescent protein. So as that particular gene, as it is not as the repressors for that particular gene kind of go away, die off, or not expressed, that gene starts to express itself more, and it in turn creates more of that green fluorescent protein, which glows. As it gets repressed, it creates less of it, and the protein over time degrades naturally, and so the bacteria glows less. And so, in essence, you get a bacteria, or a bacterium, a collection of bacteria, that sort of have this ebb and flow of glowing. Now, granted, it's not a perfect cycle, so over time you're going to have more fluorescent protein left over that hasn't decayed yet. It's going to sort of build up and create a higher baseline. But by and large, you do have a very clear cycle to how it glows. In addition, there are other things like sensors for light, dark, heat, and cold. I mean, if we as humans can detect light and dark and heat and cold, which we can, those mechanisms exist, and they exist in simpler life forms as well, being able to detect light and dark and temperature change and things like that. Repressors and activators can act as switches and logic gates. There's intracellular communication with quorum sensing, and fluorescent and luminescent proteins kind of allow output, so we can see results. If it's glowing when it's supposed to be glowing, we know that something's working, probably. Then we got to the synthetic biology contest, which is to construct a bacteria that acts as a finite state machine. So a finite state machine is essentially just a machine that detects an input and changes its internal state accordingly, choosing among several finite possible states. And again, to explain that, you can think of a simplification of a computer as a finite state machine. In fact, a computer is technically a finite state machine. It just has a lot of possible states, including ones that we aren't even necessarily aware of. There are states that we don't exactly account for, because certain aspects of programming all kind of collide and create this new hidden state that we never really planned for when we were creating maybe a flow chart of all the possible actions that a program can take. There are two different names for this. The one with the negative connotation is a bug, and the one with the positive connotation is emergent programming. Emergent programming is if you intend for new and exciting states to sort of crop up out of the natural flow of the program, and a bug is if it's undesirable behavior and you didn't anticipate it. Anyway, a finite state machine, I got to show you guys a diagram of this thing, just because this is a concept. If you ever go into like computer science or electrical engineering or anything like that, you'll have to learn about finite state machines. If you ever take discrete math, which you will if you're going into computer science or electrical engineering, or mathematics or anything like that, you'll be introduced to finite state machines. But a lot of times they're explained in much more abstract terms because, it's at a college level, and a lot of these, not all, but there are a number of college professors who can sometimes forget what it's like to be learning these concepts for the first time, and they explain them in much more complex terms than are necessary. But essentially, a finite state machine can be summed up as such. You've all played some iteration of Mario, right? Or at least are familiar with it, I would hope. So what are all the things that Mario can do? Yes, that's true. Also, interesting name. Yeah, there you go. That's a good collection of things he can do. So, let's start it out like this. Yeah. Well, basically Noah, but that's kind of the reason why I brought it up. I figured it was a very, very relatable thing. So Mario's base state is to stand still, right? If you're not pressing any buttons, if you're not doing anything, Mario is standing still. Press left or right, Mario can walk. Press a button while you're pressing left or right, and Mario can run. If you press another button, Mario can jump. I also saw ground pound. Let's put ground pound down here. We'll call it GP. He does that thing where he jumps up in the air and then he slams down and stuns or kills enemies. Let's move it up a little bit. So let's say right now that these are all the possible states that Mario can have. We'll ignore die for the moment, but these are all the possible states that Mario can have. Everything else, like picking up blocks and killing enemies and things like that, a lot of that has to do with the fact that he interacts with other things in the environment. These are not necessarily like an enemy dying. That's a state of the enemy, as opposed to a state of Mario. He doesn't enter a state where he kills an enemy. He enters into a state where he's jumping, and then because he contacts the enemy while he's jumping, that enemy enters into a state of dying. But I'm getting ahead of myself. So still, we assume no input, none whatsoever. Just like the controller's on the floor and you're staring at the screen. You press the B button, and I'm going by Super Nintendo ones here, and Mario jumps. You press left or right on the D-pad, and Mario walks. You hold down Y while Mario is walking, and Mario runs. Now, you'll notice that there is a very clear sort of sequence here. Like if we hold down Y while Mario is standing still, he won't run because we're missing an important part of the equation, right? We need to give Mario a direction to run in. Like Mario does not run from a standstill. I mean, he does run from a standstill, but he doesn't run from a standstill just by pressing Y. You need to also give it a direction. So we can assume that if we hold down Y in a direction, even if we held down Y first it takes in the direction, and then it asks if it's pressing the Y button to see if Mario's running. Now, in addition, we can also jump from any of these states by holding down B. And finally, by hitting down while we're up in the air, we can ground pound. Basically. Now, the interesting thing is, this is sort of an example of a finite state machine. However, it's incomplete because we also have to assume that you can move back in the other direction. Mario can stop running and come to a standstill. Mario can stop running and just be walking. Mario can stop walking and be still. As soon as Mario is done jumping, he might be still, he might be walking, he might be running. Ground pound, however, we can't transition back into a jump with the ground pound. As soon as you start a ground pound while you're in the air, you can't cancel it. You're committed to it, right? So at most, you'll go back to being still. Now, you might notice that pretty much every single other state goes back to still. Still can be assumed to be like our default state. If nothing else happens, Mario is still. If no input is taken, Mario is still. all other states will fall back to being still. However, we can freely move between the other ones, not 100%, but with a decent amount of mobility. So we can go from still to walk to run to jump to ground pound to still to walk to still to jump to still to walk to run to jump to walk to run to jump to walk to still, so on and so forth. All of these are different states of Mario. This is a diagram of a finite state machine. It may look complicated, but all it is simply just a diagram showing all of the possible states of a finite state machine, all of the possible states being all of these ovals with the words in them, so still, walk, run, jump, ground pound, and the ways you can move between those states. So the arrows showing how everything gets moves from one state to another. Sometimes there's a little bit of input, in this case, left, right, or the Y button, or the B button, or the down direction. In many cases, there's no input. Jump reverts back to either run, walk, or still in the absence of, anything else. In fact, by default, it reverts back to one of those states, unless you give it input of down, in which case it turns into a ground pound, and then ground pound returns to still. It's a little weird, but at the same time, it's not anything you're not familiar with, It's just thinking about the world a little differently. But anyway, a finite state machine can be anything. A finite state machine, Mario is a finite state machine because he has a number of different states that he can be in that move from one to the other. You could assume a combination lock is a finite state machine. So we have like locked, and then we have first number entered in correctly, and then second number entered in correctly, and then third number entered in correctly, and then the fourth number is also entered in to the lock correctly, but that also makes it unlock, so we can just call it unlock, and we can also spell it correctly. So you can move from lock to unlock through all these different states. Like presumably you cannot unlock a lock without entering in all of the correct numbers, so we have to move through them in order. However, at any point, you can enter in the wrong number, so you can go back to the beginning with lock. And you can also, after you've unlocked it, you can relock it, so you can head straight back to lock. In addition, you could screw up on the first one and just continually stay in the lock state. But that's kind of another thing that you can, a lot of things in life are finite state machines. Or at least they can be reduced to such in a sort of a simulation sense. So we can simulate a combination lock as being a finite state machine. We can simulate Mario being a finite state machine. We can simulate a computer being a finite state machine. We can simulate anything as being a finite state machine. And in fact, we can create finite state machines from whole cloth in order to do stuff that we need to do. Like we can create a finite state machine that, I don't know, reads the status of something and spins back out a result, or glows at the exact right time or something like that. So that this whole biology contest is to construct a bacteria that acts like a finite state machine. It takes in some input and changes its state based on that. And yes, it is the theoretical basis for computers. And now this has become a fairly large contest throughout the world, in fact. in its first year, it was a lot of very prestigious engineering schools, just in the U.S., because that's what it was based out of. And then in the second year, it started to become an international contest. In the third year, it started to spread further across the world. And in the fourth year, I believe this is outdated. I believe it's been going for more than 4 years now. I was expected to, have more than 100 schools performing. This particular example, the University of Texas, it's to build a bacterial edge detector. So an edge detector is simply just something that, like the name suggests, detects an edge. But what do I mean by that? Well, essentially, let me see here. Let me draw something with the pencil. Let me draw something straighter with the pencil. Good enough. Can I zoom in further? No, I cannot. So here's my incredibly crooked drawing. Looks kind of like New Mexico for some reason. Well, sort of like New Mexico and Arizona combined. Anyway, it's just a random shape. It's not supposed to be anything in particular. So we've got white here with the outside of the shape. We've got black on the inside here with the shape. And then you've got where the white and the black meet. So let me just go ahead and, circle an area right here. So here, the white and the black meet. That is an edge. I mean, it's just like the name suggests. I mean, you guys are familiar with the word edge, and I'm not talking about the guitars from U2. Wow, what a reference. I'm talking about an actual edge between two things. An edge detector figures out where those edges are. That is simply its job, nothing more, nothing less. It figures out where those edges are. A little bit, yeah. It does sound edgy, but yeah, I know, right? That's its entire purpose. It will not detect an edge here. It will not detect an edge here. It will not detect an edge here. Not here, not here. You get the idea. However, it will detect one here and here and here. You see what I'm saying? That's simply the purpose of it, is just to detect the difference between either light and dark or red, black, whatever, two separate things. And a bacterial edge detector in this case, they would project an image onto a petri dish coated with bacteria, and then the bacteria would glow along the boundaries between light and dark. So in this example, they got that smiley face, and it's got like the actual white part of the head, and then there's like the black eyes and the black mouth and the black background. And right where the white and the black meet, the cells would glow. As you can see here, a computer might visit each pixel in turn and check to see if it's bordered by both black and white pixels. If yes, highlight the pixel. So in this example, it's the middle pixel in this grid of 9. This one's not because they're either bordered by black or by white, or black or by white. However, this one is because there is a white pixel along one of its borders and black pixels along at least one of its other borders. So it is marked as an edge, and thus the edge detector flags it as being an edge. You can see an example of like what an edge detector does here. It's detecting all of the edges in this picture of, I guess, a Russian cathedral, and turning it into its own design. Now the bacterial edge detector, it worked pretty well. It was pretty cool. Obviously, it wasn't, um, just like this diagram here, there was actually a little bit more to it than that. But as you can see, they did get the image transferred onto bacteria. First bacterially based portrait. Yeah, it kind of does, doesn't it? Because that thing is definitely swimming with bacteria. And see, now they've actually got a couple of different strains of that bacteria. There's one that generates a replica of the photo, and there's another one that just finds the edges of that photo, um, which is, I think, pretty interesting. But as you can see here, the photo strain comes out pretty well. The edge detector strain is a little bit harder to see, but the idea is there. We got the projected mask, and then the computer sort of edge detection of it, and then the bacterial edge detection of it. And as you can see, sometimes it gets a little bit intense around the edges because the edges are, or excuse me, the points, because the edges are closer together. So it more of the bacteria luminesces. So on a Darwin. All in all, it's a pretty neat idea. The fact that you can use bacteria to represent a picture. We'll skip that. Move on to GMOs, genetically modified organisms. Now, this is a very loaded term. There is a lot of mistrust surrounding this term, and I'm not here to say like, oh, GMOs are all okay, like change your mind. No, what I'm saying is GMOs are a much broader term than people tend to attach to them in, popular society, and also to approach GMOs with a critical mind, an open mind, a thinking mind. Don't just automatically shut them down. Take a look at them. That's not to say that, the GMOs that are referred to in popular society are all safe and everything like that. I just mean uh, a lot of people that I know automatically have a reaction to this term, whether it's aversion, sometimes even revulsion, or, wholehearted acceptance. And what I'm trying to say here is to broaden your definition of this term, and also keep an open mind when approaching these in your life. So a genetically modified organism is the result of a laboratory process where genes from the DNA of one species are extracted and artificially forced into the genes of an unrelated plant or animal. The foreign genes may come from bacteria, viruses, insects, animals, or even humans. So genetically modified organisms sometimes occur in nature. We've been genetically modifying organisms long before we, were really familiarized with, well, no, we haven't been doing that since before we were aware of genes, but we've been genetically sculpting crops and stuff like that for thousands of years now. I mean, just look at what a banana used to originally be and look at it nowadays, and that's thanks to humans. Well, cloning is one of the things that can be done, at least theoretically. It's not something that we're particularly good at as people, although we've gotten better at it. as humans. Um, but yeah, Dolly the sheep. There was a clone probably about 20 years ago, Dolly, and she died prematurely. And then there was a second one. I guess it was a little bit more than 20 years ago. Either way, um, there was a second one about 15 years ago, and she's still alive today. So again, we've gotten better at it, but it's still an unclear process. Now this is genetically modified foods. So this is the popular interpretation of GMOs. So as far as this is concerned, positives of GMOs, better overall quality and taste through the modifications of foods. The flavors can be enhanced. Peppers can become spicier or sweeter. Corn can become sweeter. Difficult flavors can become more palatable. It can be more resistant to disease. So you can make them more resistant to there are diseases which only affect crops. As I'm sure you guys know, molds and stuff like that can infect crops and kill them off. Even though they don't directly kill humans, they will indirectly cause they can indirectly cause the death of a civilization. I mean, certainly less so nowadays because we have a globally interconnected economy and stuff like that. But back in the day, that was a very real risk because your entire crop could get wiped out by this disease. And if it was especially virulent, it could spread to other crops in the area, which meant that suddenly nobody could grow any food, and that's bad, very bad. And yeah, it's like a vaccine, except the vaccine is encoded into the genes instead of a shot given to the immune system because um, immune systems for plants are very different from that of other living beings, like living mobile beings. More nutritional benefits. GMO foods can have vitamins and minerals added to them through genetic modifications to provide greater nutritive benefits to those who eat them. This is especially common in developing countries that don't always have the access to needed resources. And this is also true. I mean, there are examples of GMO foods that exist, today that have things like rice with vitamin B and um, vitamin A and stuff like that in order to provide necessary nutrients to people who maybe only have access to rice, that sort of thing. Now, on the other hand, GMO crops can damage the environment. So, you can accidentally introduce an invasive species to an area that would otherwise be immune to it. So like a plant that can grow in colder conditions. It's normally a very, it's normally a very fast-growing plant that provides, that germinates a lot of seeds and stuff like that, and the plant spreads quickly. Normally it wouldn't matter in the colder climates because the colder climates are too cold for this plant, but then we genetically modify this plant to survive in the colder climates, and suddenly you have a new invasive species in this area, which can, if an invasive species gets particularly bad, it can actually decimate the existing life forms in that ecosystem. It's part of the reason of what makes it invasive. There's no economic value, so GMO foods take just as long to mature and take just as much effort to grow, meaning that there's no real economic value to growing GMO foods when compared to non-GMO foods. So yeah, we haven't really unlocked that ability for things to grow faster. We have in like a minor sense, but not enough for it to be, viable. GMO foods are better for more of their fringe benefits, like extra nutrition or extra resistance to pests or diseases and stuff like that. and a growth in allergic reactions in the general population. Now, time and time again, studies have shown that the consumption of GMO foods increases the risks of food-based allergies in people. If someone develops an allergy to soy because of GMO efforts, then if livestock eats that GMO soy as well, that person will have a high probability of an allergic reaction from eating the animal meat. This is true. So there is at least some kind of assumed correlation between the two, or explicit correlation between the two. One of the two. I'm not familiar enough with the studies to say one way or the other, and that means increased health risks for the general public, just on a daily basis, in general, which is sort of ironic considering one of the whole points of GMO foods is to increase the baseline health level of the public, and stuff like the GMO foods causing allergic reactions in people works counter to that. Anyway, that was a very short session on GMO foods. It wasn't necessarily genetically modified organisms in general. It was actually specifically GMO foods. But anyway, now we're moving on to biological clock. Biological clock. These things are fascinating. Biological clocks are innate mechanisms that control the physiological activities of an organism that change on a daily, seasonal, yearly, or other regular cycle. So you can have a biological clock that there's your circadian rhythm, which determines when you feel tired and when you feel like you should wake up. There are a number of other biological clocks that regulate various other functions in the body. There's just like when you feel hungry, when you feel more energetic than other times, how your body develops and things like that. But anyway, 3 questions on biological clocks. How do they work? How do they synchronize? And how do they translate to activity? Oh, okay. That's all there is. Let's see if this has any more information on it, because I actually think that's a pretty interesting concept. Okay, well, anyway, your circadian rhythm is one example of a biological clock. So, you might hear people going like, oh, I'm naturally a night owl, or I'm naturally a morning person, or whatever. A lot of times these things tend to be more overblown than people tend to think they are. A lot of people who say they're night owls, it's not necessarily because they're naturally more predisposed to the nighttime. They might be, but a lot of times it gets exaggerated by the fact that they're getting distracted, late in the night and stuff like that. I'm certainly one of those people who's guilty of it. My job tends to have me working very, very, very late hours, my other job. So, that also does mess things up. Stuff like that. However, the circadian rhythm is real, and it does exist, and it does influence when you would naturally prefer to wake up and things like that. Basically, it's as it's understood right now, or suspected to be, there are a couple of sort of receptors or sensory organs that exist in the general vicinity, kind of behind your eyes, sort of around the bridge of your nose, in that sort of area, that can detect natural light. When it detects natural light, it can encourage your body to wake up or stay awake. So it will, release when it's time to go to sleep, your body releases serotonin, no, melatonin, melatonin. Um, I believe it does also release some serotonin as well. And your body sort of goes through the natural process of slowing down and going into like a hibernation mode so that you can sleep. When the receptors in your head receive this natural light, they begin to reverse that progress or process and sort of force your brain to kind of bring itself up out of REM sleep and out of deep sleep and stuff like that and have you wake up. That's why you wake up after a certain period of time, even if you're tired, unless you're very, very, very tired, in which case you just, you know, stay asleep regardless. But like even if you feel like you could sleep for a couple extra hours, you might notice that sometimes you just wake up like at 9 every day, no matter what. You might notice that if you close the blinds to your room and make your room very dark, you'll wake up at like 1 o'clock in the afternoon. It's it's sort of your body's way of reacting to natural light. It's its way of knowing, of keeping track of, the day-night cycle and keeping your, well, there's that too, yeah. And there are a lot of things that go into play with it. It's not just, natural light, although that is a big motivator behind it. But yeah, if your body establishes a rhythm in the sense that like you get up every day at the same time to go to school or whatever, it will also adjust itself for that. During the school year, I work a job that has me working until sometimes 4 or 5 o'clock in the morning. Um, During the summer, I do these camps, which requires me to get up at 6 in the morning. Thankfully, I don't do both of them together at the same time. But, I'll do the camps for a few weeks at a time, and my sleep schedule will revert to such where, I'm going to bed at midnight and waking up at 6 in the morning, um, even on like the weekend and stuff like that. Yeah, exactly, exactly. It's very close to your normal wake up time anyway, because that's part of what your body does. Um, but yeah, during the school year, I've everything gets screwed up. I go to bed at like 5 or 6 in the morning, and I wake up at 1 o'clock. I don't recommend working a third shift job necessarily. It's a very draining thing. Um, that's why you guys should get into STEM jobs, so you don't have to do that. But yeah, sometimes things get off the rails and we find ourselves here. Anyway, there are a lot of things that go into the circadian rhythm. It's not just like the little receptors behind your eyes. It's also your normal sort of life schedule and um, the even a conscious um, effort towards changing your sleep schedule, and it's a number of things that all interact with one another, the background noise and things like that, literal background noise. Like if you live in a loud place at a certain time, like at 8 o'clock in the morning, everybody starts getting up and yelling at each other and stomping around, you're probably more prone to waking up at 8 o'clock anyway, because your body goes, I don't want to deal with this while I'm asleep. Um, and it tends to, scare you awake anyway or whatever. Um, but all these things work together, and your body actually, um, you know, adjusts your energy throughout the day based on that as well. I don't know why that exists there, but anyway. Moving on to an entirely different topic, DNA and HGP. Um, who remembers what DNA stands for? Oops. Nice, William. Nice. Awesome. Good work, Jacob. Yeah, so there you go. Deoxyribonucleic acid. There you go, Jean-Marc. Yeah. It's a mouthful, and it's hard to spell. Um, Thank you. Uh, that's fine. I mean, it's a big word. But what exactly is DNA? Well, here's the here's sort of the poetic way of explaining it. It's a history book, a narrative of the journey of our species through time. It's a shop manual with an incredibly detailed blueprint for building every human cell. And it's a transformative textbook of medicine with insights that will give healthcare providers immense new powers to treat, prevent, and cure disease. Like I said, the poetic way. Um, the slightly more scientific way is DNA, deoxyribonucleic acid, is a collection of proteins and genes. Well, genes that at their base are proteins that determine the behavior, the structure, and the function of each individual cell in your body. In essence, every single one of your cells in your body has DNA, and they all have their own copy of DNA. It's all the same copy, but they all have their own copy of it. So a strand of your hair will contain your DNA in it. Uh, a a flake of skin off of your foot will contain all of your DNA. Uh, One particular cell in your brain will contain all your DNA. Some of the cells that make up your stomach will contain all of your DNA. It doesn't discriminate, and those all contain your blueprint. The whole thing. They just use whatever part they need for that individual cell. But what does DNA look like? Well, I mean, it's a drawing. It doesn't look exactly like this. It's not like they've got these weird geometric shapes with the Pac-Man dots in between them and everything like that. And, it's not this garish, like blue and teal ribbon around it. However, this is the shape of DNA. The DNA does have a shape, which you can see, and it does look like this, the double helix, which is emblematic of DNA. You think of DNA and you think of the double helix. You think of the double helix, you think of DNA. Well, the double helix contains this sort of ribbon protein, which is the structure. It's just a double helix, unless I don't I don't think it's a triple helix. Triple helix is something else. But yeah, so it provides the structure of the DNA, and then there are not proteins, but I don't remember the word for it right now. Uh, there are these individual sort of I'll just call them proteins right now for lack of a better term, because I know they're called something else. And as soon as I remember what the name is, whatever. Anyway, you've got thymine, adenine, guanine, and cytosine. Um, and it's the combination of those which makes up the individual person. Now, the interesting thing is thymine and adenine will only bond with one another. That is to say, they will only be across the helix from one another. Guanine and cytosine will only bond with one another. You cannot have, a cytosine bonded with a thymine across the double helix. That just won't happen. You can't have a thymine with a guanine. You can't have an adenine with a cytosine, whatever. None of those will bond with one another. So it's all about sequences of AT and CG. What, to have them bond with ones they're not normally? Not without changing the actual structure of these, as far as I understand. Yeah. It's like saying, okay, can you force a neon atom to bond with something? No, because by default it has, you know, it's a noble gas. It sits there on its own. You could have an ion of neon, presumably, but that changes the behavior of the neon pretty dramatically, and it also is a very energy-intensive process as well. But yeah, if we did that, whew, all bets would be off as far as DNA is concerned. You'd have to rewrite the book. Um, Granted, so far, the book isn't that big, but basically everything we know would be thrown out the window. So, as it stands right now, barring further human intervention, adenine only bonds with thymine, and guanine only bonds with cytosine. And like I said, every cell in our body has the same DNA. So eye cell, lung cell, toe cell, all of these are going to have the full set of all of your DNA, all of the chromosomes and the autosomes. How much DNA is in a cell? So a genome is approximately 46 chromosomes. So these things right here, these little wiggly worm-looking things, these are all chromosomes. Chromosomes are individual sequences, well, not individual sequences, they're collections of DNA that all are combined together to make you. So a genome contains 46 chromosomes. For a normally developed human being, that number does not vary. It's always going to be 46. Now, a genome will also contain, within those 46 chromosomes, approximately 3 billion base pairs. And one base pair is 0.10th, 100th, 1000th, millionth, 10 millionth, 100 millionth, billionth, 10 billionth, 100 billionth, 3 trillionths of a meter, roughly. So that is to say, if you were to take all of your DNA and lay it out, it would be about a centimeter. No, a millimeter. A millimeter, roughly-ish. But it would be visible. That being said, DNA sequences in any two people are 99.9% identical. Only 0.1% is unique. That still means, though, that is 300 million base pairs differ. That's a lot. Still a lot. Like that's, no, 0.1%, not, so that's 3 million. 3 million, still a lot. And that helps to explain sort of the difference in people in general, because if you have 3 million slots with which you can slot in different combinations of things, either AT or CG, and you can have it be like AT, 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 CG, AT, CG, CG, AT, or you can be CG, AT, AT, CG, whatever. It's any combination of those two base pairs. Well, the possibilities are ridiculous. One might say absurdly high. But anyway, that does it for today. I got to make sure I get the poll questions in before I head off to my high school class. And we'll go ahead and do those and do a real quick question and answer time in case you guys have any, and then otherwise I will go ahead and get prepared for my high school class, and we'll meet again on Monday. So have a wonderful weekend, and we'll see you on Monday.